Welcome, everybody. I'm David Freedberg, the director of the Italian Academy. I'll be wearing another hat later on today. It's a great pleasure for the Italian Academy to host this event on how genes, emotions, and the arts influence perception, and also to help celebrate the establishment of the ZMBBI, as we all on the inside are calling it, the Zuckerman Mind, Brain, and Behavior Institute here at Columbia. We're thrilled here at the Academy to be sharing our stage in an evening that will, with any luck, give a sense of how the arts, psychology, and neuroscience can join together in the expansion of the understanding of the human brain. I actually like to think of the Academy as having paved the way for the kinds of serious cross-disciplinary endeavor that the ZMBBI will eventually enable. Indeed, even before the ZMBBI was a twinkle in the eye of our colleagues at Columbia. We at the Academy established our Arts and Neurosciences program in 2001, brought many brilliant young researchers to work in the labs of our most distinguished colleagues in neuroscience, and we continue to run our exceptionally well-attended, just as this event happily is this evening, annual neuroscience conference, mostly dedicated to showing how much the humanities and social sciences stand to benefit from an understanding of the neural substrates of culture and behavior. But now, what was a dream has become true with the foundation of the ZMBBI and with the unparalleled concentration at Columbia of some of the top neuroscientists in the world. Some of that talent is represented here today in the shape of our moderator and introducer, Mike Shadlen. So that's Mike Shadlen, not me. But let me just introduce our other participants this evening very briefly. First, Kevin Ochsner, Associate Professor of Psychology, has shaped the field of social cognitive neuroscience. Using brain imaging techniques, he studies how healthy individuals, as well as people with clinical disorders, regulate emotions, display empathy, and exhibit self-control. This will be important for my own talk because I think there will not be much self-control in my assessment of how people respond to pictures. Francis Champagne, Associate Professor of Psychology, is a pioneer in the field of behavioral epigenetics. Most of you now know what epigenetics is, but if you don't, she will explain it to you in a moment. Her research focuses on how maternal behavior can shape that of the offspring by changing gene expression patterns in specific brain regions. So I'm going to be interested in this to see how people respond differently to the artifacts of culture. And finally, Mike Shadlen just arrived at Columbia, and a great acquisition, as I mentioned earlier, for ZMBBI, was the first to apply neuroscience to studying decision-making by measuring how individual neurons make decisions that affect behavior. He focuses on areas of the brain that integrate sensory input and send information to guide motor output. This is really critical to everything we will be doing this evening, I think, at least. And the people in his lab have advanced our understanding of how perceptions are translated into actions in the brain, and therefore, presumably, in the body as well. So I'm sure that he, like Kevin, will have plenty to correct and criticize in what I, the sole humanist, have to say. And I hope that Francis at least will be a bit on my side, but we will see, on va voir. So Mike, I'm going to turn the floor over now to Mike Shadlen, who will be our timekeeper, gatekeeper, and generally responsible for the maintenance of order. Welcome again, and here I give you Mike.
Thanks, David. Well, welcome everybody. It's a pleasure to be here and to introduce this distinguished panel and to welcome you to this beautiful place. Now, I thought I would tell you, share with you some of the excitement about the Zuckerman Mind, Brain, Behavior Initiative, or Institute we now call it. And I think one way I can share this with you is to tell you why I left this very beautiful place. That's Mount Rainier, a view of it from Seattle. And I left very recently to join this, the MBBI. Because, as David mentions, I study how the brain makes decisions, which I see as a window into cognition. and ultimately a guide to understanding disorders of cognition. And to do that, we need to have interactions across what used to be very hard boundaries in the neuroscience that separated computation from systems, from molecules, from circuits, and that is all going to be taken apart. So here we go. This is my lab now, you see? Gorgeous, right? You wonder why would anyone leave a beautiful place like Seattle for that? And the reason why is because this Jerome Green beautiful building is going to house all of this neuroscience, and that is going to be the center of the Mind-Brain Behavior Institute. But the Mind-Brain Behavior Institute has a wider enterprise, and it includes the entire university. And that's why we're here tonight. We're really celebrating that. So let me say this. Neuroscience strives to understand the nuts and bolts of the brain, right? How it's built, how it develops, how it works, how it fails. But explaining the properties of the brain doesn't explain away those properties. So we have to resist, I believe, in the sciences, a temptation to conflate reductionist approaches to science with the eliminativism of radical reductionism. What I mean by that is that neuroscience doesn't explain away creativity, and it doesn't explain away responsibility or art or anything like that. And so herein lies the connection between the neuroscience and the academy at a larger, viewed broadly. And so I see neuroscience as providing a perspective, just a brain perspective or a modern angle on concepts derived and cultivated outside neuroscience itself. And it's a two-way exchange, a dialogue, really. So there's an obvious interplay between neuroscience and psychology, and we have that represented here. Obviously, biology, engineering, statistics, computer science, economics even these days. There's an emerging dialogue with art, philosophy, and jurisprudence. And I say emerging because the science itself is in its infancy, and the dialogue, obviously, is even newer yet. So now there's another dimension to this dialogue, and that's that the brain itself is always being formed, right? It's being formed by our experiences, by development, by schools, and by emotional experiences along the journey of life. And we're going to hear about that idea, those kinds of connections, in the work of Francis Champagne and Kevin Ochsner. Now, tonight, we're going to celebrate this kind of dialogue ourselves by having our own dialogue. I'm just going to moderate. And the panel is going to conclude a lively dialogue between the presenters themselves. I hope they'll make it lively. And this is an interactive part of science that this is another thing to include the public at large. And I mean, I see some academic colleagues here, but everyone in this in part of making part of the excitement of the MBBI. So we'll begin with our speakers, and I'll just explain the ground rules so you're all in on the game. So the first is the speakers are going to speak for 10 minutes apiece, and I've already warned them that at the end of their time I'm giving them a yellow card. It doesn't mean they've committed an egregious foul. It just means they're being warned, and then at the end of 10 minutes they will see a red card, and that means they're out of here. Okay, but they're not really out because we're going to have a panel discussion after that. All during the talks, please, I think you get cards that you can fill out your comments and questions, and those will get siphoned to me, and I won't censor them, but I'll try to integrate them into the Q&A part. And I think that will make it more efficient. But if someone wants to get up and ask a question that just you have to get out, go ahead and do it. Of course, if you ask a really bad question, I will also give you a yellow card. Okay, so there you go, and enjoy, and let's begin the evening then. And I believe, David, you're back.
Thank you, Mike. Where's my phone? Got to give me an extra minute for this to be switched on. Sorry. So I'm living in terror of this red card, or at least the yellow card, which will inhibit me. When I see the red card, I'll just stop and hope that I have said some of the things I want to say. So what I want to say is this, that when I first began studying the history of art, formalism, what one generally called art appreciation, which seemed to be detached from the human spectator, was all the rage. In fact, one was not allowed to talk about emotions at all. let alone the notion that when you see a picture or a photograph or a film, you somehow feel that your body is involved in the experience of looking at a work. You might say the same, of course, for music as well. So what was left out of the art history I knew as a young man preparing my dissertation was what I felt was emotion, bodily response. And since I had been very involved in the study of the great episodes of iconoclastic violence against images in Byzantium, and in particular in the Counter-Reformation, I thought to myself, how can one be so insensitive to so important an aspect of human behavior in the presence of pictures? So in 1989, which still seems a short while ago, but I guess it was a long time ago, I wrote a book called The Power of Images, which was really an attempt to describe the historical evidence that we have for emotional and bodily responses across history. Art historians hated it because this wasn't the kind of art history that they liked to have. They said, why don't you talk about how the eye travels from the lower left to the upper right and what kind of aesthetic pleasures you have? You know, this is the kind of notion that we got from the great German philosopher Immanuel Kant, who said, no interest in pictures, nothing related to the value that you attach to what you see. It all has to be aesthetic. And my aim was to insert or reinsert emotions and the body back into the aesthetic experience. So even then, in the mid-1980s, I kind of realized that we couldn't go ahead without understanding the neural substrate of emotional and physical responses, emotional, visceral and physical responses to works of art. So, you know, this seemed to me much more important than the whole notion of pure form, whatever pure form might be. So then, reflecting more, and I'm going to give you some examples in a moment, it occurred to me that it was precisely the kinds of emotional and visceral responses for which we have so much evidence across time, and for which most museum goers will themselves provide an account. It's precisely these kinds of response that enable us to understand better how some works of art appeal across time and across cultures. You know, why do we appreciate a Chinese landscape? Why do people from Islamic cultures even understand the emotion in a Christian religious picture? It's not because of the content of the picture, but because of one's visceral and emotional responses to it. So I'm going to give you some examples. Forgive me, those of you who've seen me use it. It's one of the great examples that I can think of. Some of you have heard me talk about this before. This is a painting from mid-1430s, a great altarpiece by Roger van der Weyden. So it's in the Prado now. When you go to the Prado, great crowds of people assemble before it. It's not because they know something necessarily about the fact that this is the lamentation of Christ. You don't need to participate in Christian culture to have a sense of the pervading sadness in the picture, to have a sense of the slump of Christ's body, to have a sense of the emulative slump of the Virgin's body that you see, or that woman's body, shall we say. Let me show you just a detail. You know, look at those faces. Look at the attention to the corrugator muscles. Look at the superciliary muscles in the face of the guy who turns out to be John the Evangelist. The pressing of the hands to the eyes, obviously to wipe a tear. Look at the face of the Virgin. Those of you at the back can't see, but those of you in front can see the tears that drop from her eyes. These are all essential aspects of the work. The effect of the work, even in the 1420s, depended on precisely this kind of involvement in the picture that was required of viewers, even in the theory of the early 15th century. In fact, this was a work that was commissioned to celebrate the compassion of the Virgin. When you think about compassion, you think about the notion, in fact, it was related to the institution of the Feast of the Compassio Virginis, for the compassion, the fellows, the suffering with, the way in which the Virgin suffers with her Father. So, you know, this seemed to me a perfect exemplification of the problem of empathy. And when I started thinking about the problem of empathy, you must remember in the mid-1980s, people were a little against the notion that one could say anything scientific about the emotions. The emotions were supposed to be too irregular. There was Paul Ekman, who had a kind of basic categorization of emotion. But people didn't think it could be studied on any kind of transcultural basis. So I became interested in the problem of empathy, and it was about this time that I heard of the discoveries of the great team of neuroscientists in Parma who had discovered mirror neurons in the late 80s, early 90s. And I got together with them, and you will remember that mirror neurons were those neurons in the premotor cortex which fired in the same way in the view of an observer, in the first instance of an observing monkey, as fired in the premotor cortex of the monkey executing the action. And there, it seemed to me, you had it. I mean, this is a simplistic account of a long trajectory. We feel the slump in the body, in our own bodies. When we see people making a gesture out there, we feel this in ourselves. And finally, we had some kind of neural account of what was going on in our brains, in the brains of observers, that emulated or somehow simulated, well, actually emulated in neuronal terms, the actions in a premotor cortex. I loved this. It seemed to give me an account for what you might call simulated behavior. Now, even more in this picture, let me show you, look at that. When you see that wound in the hand of Christ, you kind of start back. And so I thought, what is going on in our own bodies? Why do we get this kind of frisson of response? And I soon discovered, again, there was a kind of neural account of this, which to some extent emerged from pre-epileptic surgery, pre-epileptic testing, that when you see with your eyes a wound like this, your secondary somatosensory cortex is also activated, which produces a kind of frisson in the body. I then became very obsessed with the notion of, is this frisson local or is it general? So look at this. Here this is. Bang. Some of you may recognize this as the nail in the hand of Christ in the great Isenheim altarpiece. And I realized at that point, when I reflected on this thing, this pounding in the hand of a nail and the kind of frisson it evokes, that one had to attend to this kind of response very seriously, because when I took my daughter of 3 as a babe in arms to Isenheim with me to look at this altarpiece, she saw it wasn't just that thing, she saw the bloodied, some of you may remember, bloodied, pustulant body of Christ with tears in his flesh. I took my daughter into this room and she turned her head away instantly from the horrifying sight. This was the kind of experience which I believe we need to attend to, and which I believe we are now in reach of achieving when we look at works of art as a result of the new cognitive neurosciences. So why are we going backwards? Good. So my mirror research also made me wonder about this question, and then I will conclude with he's going to show me his yellow. I've got 2 minutes left. Concertina the rest of my talk. I became very interested in the sense that we have when we look at lamentations over the dead body of Christ, that somehow our hands are upraised. And then I asked myself another question, which has not yet been addressed, I think, sufficiently either by the psychologists, a challenge to Kevin, or by the neuroscientists. What is the link between these kinds of formulae for suffering, as A.B. Warburg would have called it, the great German art historian, pathos formulae? What is the link between particular actions and particular forms of feeling? You can register her distress straight away, as indeed you register the shock in Goya's great 12th of May, 1808. These are similar gestures. So what is the link? The challenge for neuroscientists will be to establish more clearly the relationship between motor circuits, I believe, and limbic circuits of the brain. That's a task which we won't elucidate this afternoon, but that's a task that lies ahead of us. So I have one more image to show you, because Kevin will want to talk about fear. When you see Caravaggio's great head of Medusa, look at that, you start back. It's the combination of the snakes which elicit fear, and it's a combination of the snakes and this buckle expression. You know that no one could fail to have a sense of fear, which is perfectly fitting with the image of the Medusa. I show you my final image. Here in Rubens's head of Medusa, you have a feeling not only of fear, but also of disgust. And the new neuroscience of emotion has indicated sufficiently clearly that when you look at an image that provokes disgust in you, the same parts of your brain, in this case the insula, previously the amygdala, will fire, as would fire if you were faced with this image, with this reality yourself. Interestingly, this is a picture, as in the case of Leonardo, which was kept behind a curtain, and when visitors came to look at this, the owner, we know about the owner of this painting by Rubens, would whip away the curtain and spectators would start back in horror. We now know what happens in the brain. I'm going to turn you over to Kevin Ochsner, who will pursue this matter of emotion and the way it's modulated by our experience, culture, and health or not. Thank you.
Thank you, David. That was lovely, especially the head of lettuce with anchovies. Yeah, so, and now we'll turn the podium over to Kevin Ochsner, who's going to tell us about how emotions actually work in the brain and lead us to understand our lives and perception, right? Great, Kevin, take it away.
Okay, so can everyone hear me? So today I'd like to tell you about the relationship between emotion, meaning, and the brain. And to begin, what I'd like to do is pose a simple thought experiment. Imagine what it would mean to go on a honeymoon, to give birth to a child or become a father, to eat your favorite chocolate, to glimpse a great work of art, without the capacity to experience love, joy, awe, and various other attendant positive emotions. Now imagine what it'd be like to experience the loss of a close family member, to get in an argument, to go to war, to be attacked by a nasty dog, without the capacity to experience sadness, anger, and fear. If you're like me, you have the intuition that these experiences would be but pale, pallid imitations of their former emotional selves. And that intuition has in fact guided a great deal of research that suggests that emotions are the map and compass that guide us to think, feel, and act in our everyday lives. This intuition was also shared by a group of researchers in the 1960s who sought to quantify the extent to which emotion and other factors contribute to the meanings we ascribe to people, places, and things. So they had large groups of people, like all of you, rate all manner of people, places, and things on all different kinds of dimensions that we could use to describe their meaning. When they boiled it all down statistically, they found that fully 2/3 of the meanings we ascribe are the positive or negative valence we assign to things, or whether they're simply good or bad for us. Now, if that's true, we would hope that natural selection has equipped us with brain systems whose job it is to quickly and efficiently determine whether things are positive or negative. And luckily, that is in fact the case. While there are various systems that do this, I'm going to focus on two to begin with. The first is the amygdala. It's an almond-shaped and sized cluster of nuclei buried beneath the temporal bones of your skull and the middle portion of your temporal lobe. The other is the ventral or bottom portions of a brain structure called the striatum, which would look striped with these little cross hatchings here if you were to see it in real life. And those two structures together perform what I'll call a core evaluation function for us, determining positivity or negativity. To give you a flavor for how that happens from research in my own lab, imagine that I showed you a picture of a nasty attack dog like this one. To the extent that you show a strong amygdala response, you would show a stronger negative affective experience. Conversely, if we showed you something yummy and appetitive like this, you may want to eat that bar of chocolate right now. If you haven't had dinner, don't. It will spoil your appetite. The extent to which you crave eating that chocolate right now will correlate with the strength of response in this ventral striatum region. More craving produces more activity. Now, in general, these brain systems are important for telling us what things are effectively salient, potential threats, and potential rewards. But I'll focus primarily on the amygdala, which you can see highlighted here in this portion, or this brain image, which you can imagine you would see if you took off the left half of my brain and looked in on the middle portion of the temporal lobe on the right-hand side. Now, it's not simply the case that our emotional reactions are driven willy-nilly from the bottom up by perceptions of intrinsically good or bad things in the world. In fact, we can learn to contextually evaluate or place those responses in a context. And there's a region a bit newer in evolutionary terms known as the ventral medial prefrontal cortex. It lies right behind the orbital bones of your eyes, right here. It's richly interconnected with the amygdala. And it's particularly important for putting the brakes on responses that the amygdala might cue up if they're not appropriate in the current context. So for example, if you are an individual who shows a strong response here when viewing this image, your negative affective response is low. If you show a weak response in this brain region, your negative affective response is high. Why might that be the case? Well, it may be that over time you've learned that for a German Shepherd, their bark is worse than their bite. And in fact, they don't really pose that much of a threat. And individuals who don't find stimuli like these threatening will show a strong response here as if it's putting the brakes on what the amygdala would otherwise tell you is a potentially threatening situation. And you can think of that being the job of this ventral medial frontal region more generally. Now, as humans, we don't simply have affective responses that we can put the brakes on sometimes and let go in others. We have the capacity to write sonnets, to talk to our therapist, to share our worries and concerns with others. And this capacity to conceptualize our emotion has been strongly associated with an even newer brain region, one of the evolutionarily newest and most differentiated in humans of all brain regions, known as the dorsal medial prefrontal cortex. Now, if you came into an experiment in my laboratory and I simply asked you to introspect and tell us how you feel when you see an image like this brain region would be essential for formulating that response in linguistic and conceptual terms. If instead I showed you a relatively neutral image of a ostensibly friendly-looking German Shepherd, but you started imagining it might have negative intentions, and in fact that scares you because you've had bad encounters with German Shepherds, elaborating the concept of a potentially scary dog would in fact turn on this brain region, and to the extent that you do that, you increase your negative affective response. In this case, your affective response is driven from your concept of the dog being scary rather than the actual presence of an attack dog out there in the world. And of course, you can turn this facility to people outside yourself, trying to interpret how these people are feeling in these images. For example, here, trying to determine whether that woman is more frustrated and excited than she is angry. Now, you may wonder, is it always so simple? Do these brain regions quickly and fairly effortlessly determine the affective qualities and meanings of our lives? And the answer, of course, is no. So on the one hand, sometimes what you are feeling isn't clear. So if you take this new father, for example, and a friend asks him, how are you doing? What's it like to have a young child? There are any number of emotions that are swimming around inside him. Which one of these most aptly describes his experience at a given moment in time? Which one should he express to his wife? Awe. Which one does he express to his friends? Anxiety. I have 2 toddlers. So it turns out to solve this kind of emotional problem, you call into play an additional part of your frontal lobe, which lies behind the frontal bones of your skull, behind your forehead. The lateral prefrontal regions highlighted here. Now, historically, we've thought of these brain regions as important for dry, cold cognitive abilities, reasoning, logic, problem solving, executive control, if you've heard that term. Here, the problem that they're turning their attention to is not a simple logic problem or something on the GRE, but rather the problem of disambiguating your own emotional response. Now, of course, other people's feelings aren't always clear either. Consider these 2 images. Here, there's a smiling fellow you might think you should approach. Here's someone who looks potentially scary and you should stay away. But we're capable of conceptualizing the conflict between nonverbal facial expressions and the situation in which they're embedded. A used car salesman isn't happy to see you, he's trying to manipulate you. A politician like Jim Webb, who's just had a major political victory, is exultant, not threatening. And we can conceptualize this and resolve the apparent conflicts of this sort all the time. This lateral prefrontal region is essential for resolving those conflicts. And then a final way in which we use this capacity for controlled cognitive processing in the service of emotion is when we need to transform the nature of the emotional response itself, volitionally, effortfully taking control and changing the trajectory of what we're feeling. So let's revisit our friend, the barking dog. Let's imagine that that is the beloved longtime pet of someone you've just begun dating. And you don't want to let on that this is in fact very scary for you. And you choose to reappraise the meaning of the dog's barking. You say to yourself, this dog doesn't intend harm, it's just marking its territory. Really, he's scared of me, at least as much as I'm scared of him. And what you'd find in the act of doing that is that these lateral prefrontal regions are helping you inhibit or turn off the amygdala's response as you formulate this alternative narrative for the meaning of the situation. But if you get caught up in doing the reverse, imagining the dog really wants to hurt you, it might break its chain, it could cause permanent injury, you've got a bum ankle, you can't get away, she's going to dump you, oh my God. What happens, in fact, is that these prefrontal regions are failing to solve the problem appropriately, and they're in fact making you worry and ruminate and boosting the amygdala's response, adding cognitive insult to perceived injury, if you will. Okay, so where does this leave us in the end? What's the upshot for how we think about emotion, meaning, and the brain? Well, one thing I can say is that we're getting closer to associating specific functions, affective functions, ranging from quick assessments of valence and intensity of stimuli in the world to elaborate conceptual abilities to write sonnets and express our feelings in words. And we also have regions involved in control that help us disambiguate our feelings and those of others and change their very nature. But what I haven't told you is the magic of how all of these brain regions talk to one another in real time to construct the nuances and flavors of our emotion as they happen from moment to moment. The reason is that's at the very bleeding edge of our understanding of emotion circuitry in the brain. And when I daydream about what our future understandings might look like, I'm guided by a simple analogy. Emotion is to the brain as music is to the instruments that produce it. The simple idea then is that you can think of brain regions as playing emotional notes that combine in various ways. And the emotions we experience are different kinds of music and different pieces of music whose meaning can't be understood as the simple sum of the emotional notes from which they arise. The question I think that's going to face us in the next couple of decades is how do these notes give rise to emotion? In terms of their tempo, their timing, and so on. How do prefrontal regions that can act as a conductor that tells these instruments, if you will, when to come in, when to play their notes, when to change their tempo and their volume? How does that all come together? And the hope, of course, is that the Zuckerman Mind-Brain Behavior Institute will help us do exactly that. Thank you.
Perfect. All right, thank you very much, Kevin. That was lovely. And our final expert is Francis Champagne, and is going to tell us, I think, a little bit about epigenetics and the developing brain and sort of how we become who we are and appreciate what we appreciate.
No. There we go. Okay, so one of the questions that really motivates my research is, why are we unique? So you've heard from David and Kevin a lot about the experiences that we have and the emotions that we have, but certainly individuals differ in their reactions to Caravaggio's Medusa or their reaction to a very frightening stimuli. And we're all curious about where that uniqueness comes from. Why am I the way I am? And where are the origins of these differences? And historically, this question leads us to nature versus nurture. Am I unique because of the unique genes within myself, the unique sequences of DNA that build my character, or am I unique because of the unique experiences that I have across my lifespan, particularly those experiences that are occurring very early on in development? And so nature and nurture have certainly been fighting it out for many decades. And what we're trying to find is a resolution to that battle and how it is that both of these influences come to shape our being. Now, when it comes to DNA and how that might shape our brain, we have a slight idea of how that might work. We know what DNA does. It produces mRNA. That produces protein. The proteins go and do things within our brain and activate circuits and change our ventral striatum or our amygdala. The question is, how could the environment also shape the brain? Through which mechanisms can this be achieved? Now, it's certainly the case that the environment can do this. And so decades of research and certainly anecdotes across the centuries have shown that our early life experiences can have a very profound effect on our brain, on our physiology, and our behavior. The experience of severe stress during pregnancy can lead to many outcomes in offspring, including depression and anxiety. The experience of neglect in early infancy can likewise have a profound effect on functioning in later life. So our experiences of our nurture can have a long-term effects. The question, of course, is how? How is this achieved? And so my research is really focused on trying to get down to the biology of how the environment shapes our brain and behavior. And that led me to really go back to the question of nature and how genes actually do this. So we know a little bit about how genes work, but in fact there is a very active process through which genes come to shape our biology, and that same active process may be an explanation for how the environment comes to shape the brain. So we have our DNA, but it is compacted within our cells and must be read. So like many books in a library, our DNA, our genes, need to be read to be able to inform us, to be able to inspire us, to be able to do something within our biology. And in the recent decades, there has been increased interest in the factors that can change the activity or the reading of DNA. And these factors are referred to as epigenetic. So there are a number of ways to control whether DNA actually does something, whether genes become expressed and produce protein and have a biological effect. So here, if you can see, are some of those molecular changes. So we can change the activity of genes through changing of the proteins around the genes. So our DNA is wrapped around a cluster of histone proteins, which really allow the DNA to become compacted, but chemicals can be added to those histone proteins that change the activity of DNA, that make it less active or more active, because it makes it lesser or more likely to be read by the mechanisms within our biology that read our genes. In addition, we have a process called DNA methylation. This is where chemicals get bound onto our DNA. So it doesn't mutate our DNA, but now we have these chemical tags within our DNA that give it kind of a unique character. So we have our DNA sequence, it's unaltered, but now we have this extra information, this epigenetic information that has become layered within our DNA. Now, this process of changing factors around the DNA, globally classed as epigenetic, leads to the turning on or turning off of genes. And this process is so incredibly important to our biology that if we could not do this, we would not survive, right? We would not be able to develop beyond very early embryonic stages and survive during postnatal development. We need to be able to shut genes on and off in a very, very refined temporal pattern. Now, the question is, okay, great, we have a way to control our genes, and there are many, many ways in which this can be achieved. What can control those factors that can control our genes? Well, the fact is almost everything induces an epigenetic effect. Many of the experiences that we have across the lifespan can induce this. So currently, there's a great deal of interest in how, for example, smoking, drug use, exposure to pesticides, exposure to nutrition, exposure to hormones can induce an epigenetic change. Now, these epigenetic changes, like most changes in the brain, are more malleable early on in development, so during prenatal and postnatal development, exposure to hormones and variation in nutrition can have a profound effect on epigenetic landscapes, but certainly across the lifespan, and these epigenetic factors can be modifiable by experience. But these factors, you know, where we're exposed to pesticides, it's not that surprising that it should have a profound biological effect. But the truth of the matter is that social experiences and the experiences that we have with individuals, particularly individuals when we're developing, have a profound epigenetic effect. So emerging evidence shows that the early experience of abuse, neglect, stress, or variation in the nurturing environment can have a profound epigenetic effect. And so epigenetics and variation within our epigenome is one way for a multitude of experiences, both social and non-social, to become incorporated into our biology, leading to long-term changes in behavior. Now, one of the specific experiences that motivates a lot of my work is how variation in maternal care can induce epigenetic change. So, using laboratory studies with rodent models in which we have very limited genetic variation, because that genetic variation can be very pesky when you're studying epigenetics, we found that, you know, despite having very little genetic variation, we have these animals with a rich variation in behavior. Some are more reactive to stressors, more emotional. Some are more rewarded by chocolate and other rewards. How can this be? And so when we looked at the early maternal behavior being experienced by these individuals, we found that, in fact, those individual rats and mice that are getting lots of maternal care during early development, that leads to a very precise epigenetic landscape. In the case of the stress response, high levels of maternal care leads to a persistent activation of genes that allow you to turn your stress response off. So having a stress response is a wonderful thing because you need to react to the world, and that's what our stress system does. But having a prolonged stress response is not necessarily beneficial. So we need to be able to react to stressors but turn that stress response off. And what maternal care does is activate genes that allow us to self-regulate, to change our stress reactivity once it's launched. Those individuals who are capable of doing this can cope better in a stressful environment by shutting down their stress reaction. Maternal behavior also shapes reward centers and maternal behavior centers in the offspring brain. And so mothers engaged in more care towards their offspring, those offspring have long-term changes in their own maternal behavior. And this is through epigenetic changes in genes responsible for reproductive behavior, such as the estrogen receptor. Now, experiences within your own lifespan certainly can have an epigenetic consequence, and that's well and good, and can have both transient and dynamic effects, but also long and term persistent effects on the way you function, on the way you perceive the world. But interestingly, this has opened up a new window into insights into how factors and our experiences can be transmitted across generations. Now, females that experience high levels of maternal care tend to be high in maternal care themselves, as do their offspring and grand offspring. And so we have roots through which factors such as stress, social experiences, and exposure to a variety of environmental conditions can be transmitted across generations. But interestingly, epigenetic variation can also be inherited, much like genetic information. And so there is emerging work that a number of life experiences not only affect you, but can affect your offspring and grand offspring. Of course, within the context of all of the pollution and nutritional variation and social world that we're experiencing, this is a profound realization and is really changing the way we think about the origins of our unique characteristics. And so finally, I'll end with a note that one thing that I really like about epigenetics and studying the brain is the fact that there is both plasticity and stability. So epigenetic mechanisms doesn't mean that you are set for life, that you are going to be stressed, and that's it for you. These are changeable mechanisms. So this is a wonderful avenue for therapeutic intervention where we can change the epigenetics and change your developmental time course. And I'll end there. Thank you.
All right. Thank you very much, Francis, and everyone else. I think at this point, I'd like to remind the audience to fill out cards if you have questions. And I'm going to invite the three experts up onto the stage, and we'll have a panel discussion before opening the questions to the rest of you.
Sit in the middle. Oh, I'm there. Okay. Oh, whatever. Okay. Oh, I see.
Oh, okay. Did one of you leave a watch up here? All right, well, do you have questions for each other? Because if not, I'll get you going.
Why don't you get us going?
All right, so I'd like to know, when we react to art, and there's differences in the room as to how people reacted, do you think that this is something that began with the way our mothers treated us, with some part of the brain, or something that we really can't explain even at the level of the brain that we just have to strive for?
So can I jump in here?
Yes.
What to do with art. So actually, I noticed that Francis, I was really interested and happy, although I'm going to argue with her about it, that Francis says we all have different responses to the Caravaggio. So this is, in a way, I began my interest in neuroscience and cognitive neuroscience precisely in frustration with this attitude, in the sense that it's obvious that we all have different responses to works of art. I spent a large part of my early life going to archives and saying these people had this attitude and these people paid so much for works. But what really interested me in the end, this is why I'd like to ask you perhaps both to elaborate on this, was the fact that when you see somebody open mouth like that, I mean, you gave a good example, but there's no other context. You see this open mouth, you see these snakes, and I can bet you that when you look at your fMRI scan, you will see amygdala reacting, boom, straight away. It's not going to be eliminated unless you have some kind of deficit. So that's really what seemed to me the great puzzle for our time. Not to look at differences. We all know that we're different. But to look at those operations in the brain which are identical across the species. So in a way, what I'm not denying is local modulation. What I'm interested in a way, is what precedes context, what precedes local modulation. Everyone gets a fright, whatever culture they come from, when you see it, teeth exposed, mouth open, and snakes in most cultures. So, the real question is, what can we get below difference, rather than just look at individual differences?
Francis?
Well, I think it depends on, what drives you and what drives your interests. I certainly appreciate that we would, as a group, we would have a lot of common reactions to stimuli. But for me, I like that noise. I like the variation that's going on with individuals, and for a lot of studies, that noise is dismissed, and it's just the background variation that exists within individuals. But I would want to know why someone has better breaks in their brain than another individual, because it's important, right? Because it controls how we respond to the world. And it's interesting when it comes to fear, because there's been this whole history of the innate fear response. This is something that we are born with, and it's not something that we learn. And this is something that gets you into all sorts of troubles when you talk about anything being innate. Yeah. But certainly, as you say, we do have, in a sense, a gut reaction to individuals. And I'd be interested in the process through which that develops. If you were deprived of the normal kind of social, emotional context that we tend to have in development, would we still develop this reaction to stimuli in the same way? So for me, the variation is the interesting thing.
Well, maybe it's just because I'm sitting in the middle, but I think they're both right. I think you certainly want to know what the core processes are that operate regardless of individual differences, but then you also want to know what the sources of individual difference are. And at least for the kind of research I do, where you're studying the human brain in healthy and clinically disordered individuals, there's a distinction between, we do better with addressing your question than your question, because there isn't really a neuroscience of mental content right now. So I could show you the Caravaggio image or the scary barking dog, or give you an electric shock, and the amygdala would respond the same way, let's say, in all three of them. But that's not where you'd look for the differentiation of the different qualities of the stimulus that elicited it. In the same way, we can resolve color regions of the brain that we know might be active when you think of a Christmas tree and you think of a fire hydrant, two entirely different colors, but at least at the limit of our resolution with imaging, all we see is that a color region is active. So we're getting better at doing that, but we're better at addressing this question now than that question. But they're both so important.
So are you sitting in the middle, or are you agreeing more with one of us? I can't quite
I agree with you both.
Okay.
He's exerting
No, I mean, I
himself.
think, the real question remains for me I realize now, I've thought about this for a long time, but the real question remains with all artifacts of culture. How is it that some artifacts of culture actually transcend? I mean, my colleagues in the humanities now, who have gone completely in the direction of difference, would all be appalled at this question, but I remain concerned with the question, how is it that some artifacts of culture transcend individual differences, give or take some nuances? In other words, we're living in a global world where regional differences are being eliminated, but even before that, regional differences could be subsumed across the globe, across, in a way, the species. So that, for me, is the fundamental question. In a way, the less interesting question remains the seeking of difference. And I still haven't heard quite how one relates these two levels of experience. So in other words, what I'd like to crudely call, you'll be shocked, the precognitive versus what happens with cognitive operations.
Well, I mean, there's probably two kinds of answers to that question. One is that while we may have, all of us have the capacity to, say, be afraid or be sad, and there may be some universal life situations, like the death of a close loved one, for example, that would make us all feel sadness and grief. There are many throughout our life idiosyncratic contexts that are going to lead to trigger particular emotions that may be importantly different for me than you, and that difference could lead me to end up having depression and you not, for example. And understanding the nature of those individual trajectories towards one type of life outcome versus another is incredibly important. And it may be that some of the processes are the same, but the inputs to them, the triggers for them, are fundamentally different. And there are different parts of the brain that represent those triggers than are the sort of final common pathways to triggering the emotional response.
So is the emotional response something that lies at the sort of core of what allows us to be human? Or do you think of the emotional response as the ultimate completion of this very highly intellectual, in some sense, act of evaluating the content of an image?
I don't see them as either or. I mean, I think emotion, the human's capacity for emotion is probably different than other species on the planet, precisely because we can be so conceptual. We can have experiences like nostalgia, where we're happy and sad about something in the past at the same time. I love that emotion. I love mixed emotions. I love having combinations of emotions at the same time that you can reflect inwards upon and try to understand the nature of, and then express them to others. To me, there's a level of richness and complexity that we have that's maybe defining of human emotion, but not of emotion in general. And I think that's one of the reasons I presented neuroscience data on emotion in the way that I did. The idea that there's a gradient of complexity of the kinds of emotions that can be driven by evolutionarily old subcortical structures that are the most simple and basic, up to emotional responses that we can appreciate in terms of complexity and nuance with a fine grain of appreciation and experience that are driven by portions of the brain that are evolutionarily the most new and most differentiated. Like that dorsal medial frontal region, there's some work that suggests that it's the most differentiated portion of the frontal lobe in Homo sapiens sapiens us as compared to all other primates. So there's something to that, and the qualities of emotional experience that it supports.
I mean, I want to let Francis come in, but briefly I want to just, again, disagree with Kevin here, if I might.
I'm having an emotional experience.
It's an emotional experience. No, I mean, you know, this is a slip here. I mean, I think it's too much on the high level, and I'm inclining to what I think Mike is sympathetic to sort of explore slightly lower level areas of experience. I'm not interested, for example, I wouldn't deny for 10 seconds that, you know, the relationship between situations and emotions cannot be universal. But what I'm interested in, what I think we need to discuss is what actually happens when you feel an emotion, irrespective of the situation which produces it. So that's what we have to study, which will then illuminate different responses to particular objects. So, you know, it's when somebody feels disgust or something, we can be fairly clear that the mechanisms involved are pretty consistent across the species. That's the interesting thing, and that's what I think we're learning as much from neurosciences as the ways in which they're modulated by experience or epigenetic variation. So you can talk about the epigenetic variation.
I think it's exactly what Francis is talking about, really, right?
Yeah, well, you know, just talking about the cross-species comparison, I think is really interesting, and your point's well taken that we do need a better marker of when someone's in a particular emotional state. You know, in neuroimaging studies we have, or any studies, we have images that we use to that we have characterized as happy, angry, distressed, but trying to find the neurobiological and physiological correlates to that is actually very challenging. And I was involved in a study on dog emotion and rating dog emotional states, but we don't know anything about the internal state of an animal when it's in a particular emotional state.
So I would argue that the core function that allows us to even create art is this sense of an emulation, I think you used the word, or an empathy for the other. And I wouldn't be surprised if the epigenetic mechanisms that occur early in life sort of predispose our brains to being able to experiencing through the eyes of another, watching their actions, inferring their intentions, watching their emotions, feeling them ourselves, seeing them see something else and feel our own repulsion to the same thing. This is what allowed us as societies to expand beyond kinship to kithship, and it allowed us to create great art. So I think it's sort of a core function of the brain, and probably establish the epigenetic mechanisms that occur in your photos between mom and child, and we're learning more and more about them.
Yes, definitely, and there's actually a lot of work now looking at how variation in maternal care in humans can shape these empathic responses, and there's great concern that if we engage in certain types of mother-infant or parent-infant interactions, that we would lead to a trajectory away from this kind of emotive, empathic type of response. So I think it is something that develops over time in response to the strong social bonds that are formed early in life, because that's where individuals learn about emotional states in others and how that affects them as well as other individuals.
So Kevin, I was kind of in a way making an allusion to David's reliance on the mirror system, and do you think that's of any relevance to your work?
So there is a whole other line of research that we've done that's on empathy that looks at the way in which the brain system I referred to as important for conceptual evaluation. That's a term that I like to use in the context of emotion, although people use a different terminology in other contexts, like theory of mind. If you ever come across that idea, it's the idea that the developing child develops the kind of capacity that Mike's been talking about to understand that other people have beliefs and desires and feelings different than their own. They have a theory that other people have a mind, and that's why they're acting the way that they are. You have internal goals and beliefs and desires that lead you to act towards me in a particular way. That's what drives you to eat, and so on and so forth. That brain region is particularly important for that, and that's a big contributor to empathy. The other piece is something that David mentioned. it goes by the popular term, the mirror neuron system, but the basic idea is that you have motor regions in your brain, in the frontal lobe and the parietal lobe, that are important for programming goal-directed actions. So if I reach out and I pick up this glass, I have the goal perhaps to drink, and there are motor actions that go along with that. It turns out that if I see David reach out to grasp his glass and start to drink, the same motor regions, or at least a portion of them, will become active, as if my brain is simulating what it would be like to pick up the glass and drink it, even though David's doing it and I'm not. And the thought is that that gives us some intuitive, implicit understanding of what it's like to be the person I see acting. The regions for action in my own brain are active when I observe someone else acting. And we've done some work that shows that those motor regions, along with this medial frontal region for conceptualizing thoughts, feelings, and desires, work hand in hand to support your ability to accurately understand other people's emotions, to take their perspective, if you will, and see the world through their eyes and understand how it is that they're feeling. So I think they're constantly working back and forth, and neither one turns out to be sufficient. And actually, you'll often see in the scientific literature, there are debates that evolve where one camp has studied one brain system, and they'll say empathy is always that. And a few years ago, David put me in a debate with one of the progenitors of the idea of the mirror neuron system, a lovely man from Italy, who has a lovely accent that makes you all the more convinced that what he's saying must be true because of the accent and his bodily gestures that he's hoping you emulate in the audience as he's talking. And I questioned whether or not the mirror neuron system was the sufficient basis for us to understand other people's emotions, as he claimed. And it's clear now it simply is not. It's important, it's a contributor, but it's not. You need this other capacity to conceptually simulate the mind of another person. It's not sufficient to have motor mimicry, full stop.
I think we have to let David respond to this.
Well, I wasn't going to. I mean, he's slightly modified or modulated his antipathy towards mirror theory on its own. But actually, I don't know whether I want to respond to this question, because then we'll get very technical indeed, although it does allow me, perhaps, this will get us a bit away, I think, from Francis, whom I wanted to bring in again, but it does allow me to ask you to reflect on the relationship between the movements one perceives and the movements one feels in one's own body and the emotions one feels. You know, when you do this, you have a kind of feeling of excitement. When you do this, wiping away tears, you have, it's a hypothesis, you have a sense of sadness. I mean, it's interesting that the bodily technique is so closely related to a felt emotion. For me, the mirror proposal actually serves to go a long way to understanding the relationship between the body and felt emotion. I've always, just going to move on to a slightly separate point, I've always preferred mirror theory to theory of mind theory, because I never know, when people talk, I'm sure there are lots of supporters of theory of mind over here, I never know what is the nature of the theory in theory of mind. This is something which has been imagined by psychologists for actually quite serious purposes, as some of you know, for the understanding of autistic behavior, for example. And it's always been unclear to me what theory of mind really is predicated on, what neural states theory of mind.
David, it encompasses all of our social cognition. No one would paint a picture if they didn't think that another agent could appreciate that picture, and that's all theory of mind is. So we're now going to turn, that was cheap, but we're now going to turn to the questions from the audience, okay? Let's see, I think I was supposed to remind you of something. Is that true? Right, no, not yet. All right, so here we go. I'm just taking these at kind of random. Okay, so how about this? Can a change, several of you asked questions about the environment, and so one of the questions is, can a change in the environment, diet, other factors, cause epigenetic changes in offspring? Francis, I think you're best situated to do
Yes, it can. So, we've known for a long time that diet, exposure to toxins, exposure to stress hormones can induce long-term changes, and now there is quite strong evidence that the level of nutrition that you have, say as a parent, can affect the offspring's epigenetic code. Now, what's I guess very interesting in this, it's fine when when the nutrition variation or toxin exposure occurs during pregnancy. Okay, great. The mother is ingesting different foods or exposed to different toxins, and there can be direct exposure to the offspring. But I think what's very interesting is that the exposures can occur preconceptually. They can occur well before you even have children, or even thinking of having children, even by your parents and grandparents. And so there's increasing evidence that nutritional exposures can have transgenerational effects, and even the nutritional level of fathers, which is something that we're studying now, can have an impact down generations through changes in DNA methylation and histone modification. So it's very, very Lamarckian and very, very different, but it's happening.
Interesting.
I wondered if Lamarck was going to make a showing. Okay, well done. I think this one might be more for Kevin, but anyone can chime in. Can someone learn to change how they react to situations, or are emotions hardwired?
You can definitely learn. I mean, there may be some types of emotional responses that are more difficult to regulate than others. So the experience of disgust, for example, is one that turns out to be somewhat difficult to change. Although there are very cute experiments that have been done where there's a particular odor that, depending on whether you believe it's smelly sweat socks or a particular kind of odorous cheese, elicits either disgust or desire. So there may...
Which, though?
It depends on what you think it is, right? What you can do is you just frame the meaning of it for people. You tell them, this is, sweat sock in a bottle, sniff, oh, disgust for the areas of the brain fire, or this is a particular kind of cheese. Oh, I might want to try that. That sounds interesting. And so, there's a limit to how much you can probably regulate particular responses, and there's a delicate interplay between the kind of strategy that's most appropriate given the type and intensity of the emotional response. Like, one great example of this is that if you're in the throes of a very strong response to a trauma, you don't want to try to get in there and verbally unpack and figure out the meaning by reappraising it, reinterpreting it, and talking it through. It turns out that's actually detrimental, because what happens is, in the act of trying to get in there and rethink this traumatic event when it's still so strong and fresh and recent, the strong affective response you have starts shaping and guiding your thinking. Instead of you're trying to make it go over here, but the emotional response is so strong that it keeps pulling you back, and you just end up elaborating the meaning of the negative event. This, in fact, happened to a lot of people after 9/11 who were in the towers. There's a whole lot of research that sparked, but had been done prior to that. But what you want to do is try to talk things through later on, not right away. So the answer is always, it depends.
All right, here's another one. It's kind of a double question here. Does the quality of a painting affect the amygdala response? Is a Caravaggio more effective than a Norman Rockwell? Or a scene, or still, I guess it says from South Park. Sorry, whoever did this, I'm botching your question. Related question, why does anyone like Jackson Pollock? That's not for me, that's the questioner, right? I feel like I have to do the David Letterman thing. Right. I think that's for you.
But
how does it make you feel?
Am I obliged to answer both those questions? I'll have a crack at it. So I'm blown away by the second question. So the first question is, I don't know, I can't speak to all the functions of the amygdala. Kevin could do this better, and Dan Salzman, who is unfortunate, I don't think he's here, Dan, he's not here, could also speak to the matter of amygdala. I don't think amygdala is much affected by the technique of a painting, unless maybe you had a lot of jagged lines going across a picture. But in the case of, what's so interesting in the case of a picture such as the one I began with, well, let's say the Caravaggio, you may admire the quality of the painting, but your attention is first caught. We know this by the Buckle expression. In other words, by the appeal to the emotion of, by the appeal to the emotions of an open mouth, which can either be a scream or it can be the exposure of the teeth. And of course, combined, here's a purely contextual thing, with the snakes, you can be fairly sure that that's what grabs your attention first. Everyone will admire the skill of a work. I do think that in a way, the effectiveness of work is often related to the maker's, let's not even call it the artist, the maker's ability to evoke the kind of emotion that she or he wishes to convey to the spectator. In other words, Rockwell probably doesn't make it to the Caravaggio level because, other than perhaps evoking the kind of nostalgia which Kevin was speaking about, it somehow doesn't really arouse the kind of strong emotional response, the kind of effective emotional response, the kind of response that the painter wanted most strongly to convey, as in the case of the Caravaggio. So I have quite a clear sort of bar here for effectiveness of pictures, which is related exactly to the issues we discussed. Jackson Pollock, I'll just say one thing. One of the interesting things about Jackson Pollock, the two interesting things which neuroscientists can help us with. The first is the way in which the direction of the strokes may activate parts of the motor system. And here I actually have a mirror theory also for Jackson Pollock. So we know how we get engaged by the sense of movement that we feel when we look at a Jackson Pollock. Another interesting thing we haven't talked about. I'm always fascinated by when we look at an abstract work, we may find that we don't know what's going on. And one of the first things we do is to try to find a face in the picture. Jackson Pollock said, no faces in my pictures. But we have this kind of physiognomic hook. We look for objects in the apparently incomprehensible. And so I think there's a lot to think about. When you next worry about a Jackson Pollock, think about those two things.
Okay, I think we have time for maybe two more. This first one, there's three questions, two of which are crossed out in this card. And whoever wrote this, you know who you are. They're excellent questions. You should be a neuroscientist if you're not already. But the last one kind of reveals that you're not, because it says, what are the long-term benefits of understanding the brain? Francis.
The long-term benefits. Well, I think the long-term benefits of understanding the brain. I think the greater understanding that we have of the brain, the greater understanding we have when brains don't work. And I think for me, it's the most important thing is about intervention treatment of individuals who are disordered in their behavior. And we as we gain more understanding of how the brain works, we can find improved techniques for repairing function of the brain when it's disordered.
Anyone else want to take a crack?
I'll just say here.
Well, I mean, I agree with that. I mean, obviously, I'm driven by that. I'm a neurologist, and I see the study of higher brain function as giving us insight into what it is about the brain that we care about and that goes awry in disorders that we might just say make us confused, whether they're autism or schizophrenia or attention deficit disorder or what have you. And so these kinds of new neuroscience of cognition, which is, we're touching on a sort of one of the most beautiful aspects of it, what makes us feel the way we do, gets to the core of what it means to be human in the sort of large integrative sense. I don't want to in any way dismiss the important research that's done on just perception per se, or movement control. I mean, we know that if you have a stroke or something, that you have trouble moving your body, that's a very devastating illness, and we need to solve those kinds of problems too, motor neuron disease and so forth. But the kinds of things we're touching on here tonight in cognition are the kinds of things that we'd like to one day be able to remedy. So
Mike, if I can just add a simple analogy to that. So it just occurred to me while both of you were talking that you could ask the same thing about any object or mechanism in the world, like what's the long-term value of knowing how a car works? Well, so long as you're just the driver of the car that always works, you don't need to know how an internal combustion engine or the suspension works. But as soon as it breaks, you need someone who knows how it works.
So I feel really bad because the last question was going to be from an actual Italian, but we don't have time for it. I'll just say that blind people also experience emotions, and it doesn't have to necessarily be through vision, but of course blind people have a panorama of the world before them, and so that's the answer to your question about stimulations for blind people. So I think at this point I'm trying to keep us on schedule here, I'm supposed to, oh my God, invite you to do something. Yes, invite you to talk to the panelists downstairs once we close. I'm going to just say a few quick closing remarks, if you don't mind. It's very hard to kind of put these three together because I thought they were just absolutely beautiful talks by the panelists. In fact, let's thank them together. And while we're on the thanking, I'd like to thank, of course, David Freedberg for sharing the Academy, the Italian Academy, and the Institute. And I'd also like to thank Abby Asher, who I think has helped organize this, and Kelly Raymall, who's standing over here. And of course, we'd like to thank the Zuckerman Mind Brain Behavior Institute. I mean, I'm very grateful to them. I mean, they are why I came to Columbia and get to meet all these amazing people and allow us to, in thinking about the brain, also in the same breath, think about great art and emotions and Lamarck. My God. Don't tell my high school biology teacher. And then, let's see, I'm supposed to remind you all that, this is Brain Awareness Week, and that is something established by the Dana Foundation. It's the Dana Alliance for Brain Initiatives, who also supports a lot of what we do here at Columbia and throughout the city. And there's something called Brainy, B-R-A-I, and then New York, kind of, Brainy. And there's all kinds of things going on all over the place. There's a website, I think it's written on the form that you came in, that tells you where you can find all kinds of events throughout the city. I would like to thank you all, the audience, for submitting all your fantastic questions. I'm really sorry I didn't get to probably only through a third of them. But of course, we'll have a reception. I do want to try my hand at trying to put together what you heard tonight in about 30 seconds. So let's, my friend Daniel Walpert, who's a neuroscientist at Cambridge, England, I always ask the question why we have a brain, and he has a pat answer to it. It's to move. He claims that the most successful organisms in the world are trees. They don't move. They don't have a brain. And many other very interesting examples. But now some may say it's to move. Others may say, like me, like it might be to make decisions or something. But maybe really it's to feel. Okay, and that's what the emotional brain is really about, this discussion. In fact, I would argue that to appreciate great art, as I mentioned to David a moment ago, we need to feel deep down, and David, talked about this, and he's evoking the mirror system, but I think it's just a much more general system than that. It's the system that allows us to have compassion at all and imagine that another agent is also experiencing something, not necessarily what we're experiencing, but something at all. And that's, I think, what theory of mind is about, which is just a catch-all term for that basic capacity to imagine that another is also experiencing the world. For my own money, that's also the nature of what gives us consciousness. It's that sense that there's a world not just experienced by me, not just my affordances of what I grab, but a world that's also experienced by another agent and therefore has existence independent of me. And I think it's a fun way to think, to put the sort of the social brain at in the front. And what we heard from Kevin was the really elaborate aspects of the brain that allow us to both not just have direct emotions, but also to contextualize them. And he gave just some fantastic examples of that. And they work the other way around as well, that if you just try to mimic, someone asked a question about acting, but if an actor tries to mimic, or even inadvertently mimics a smile, they react differently to even just neutral material put before them, okay? If you just get your head into a frown, apparently, I'm told by social psychologists, you're more likely to react negatively to something than if you had something in your mouth that made the muscles imitate smile. So it goes both ways. And then back to Francis, I mean, the establishment in our bodies through the power of epigenetics, which we're just beginning to touch on, right? It's in its infancy. We're just beginning to realize that through maternal-child interactions, through our early experiences, and sadly probably through our experience with toxins, we change just the very nature of what we are. Not as dramatic as necessarily a cancer, but the difference between the way our brains function. And I think we put all those things together and we think about a brain that nurtures and a brain that accepts nurturing and sort of sees in others the possibility of nurturing, wants to nurture, and the extension of nurturing is not just to your child, but in a sense to life and to embrace it with others. And that's what we're doing here as we celebrate this exercise and enterprise to understand, appreciate, really celebrate the mind in all of its glory, ultimately perhaps to fix broken minds in disease, but also just to appreciate all that is around us that is the human condition. So on that, let's thank the speakers one last time and then go downstairs.